Oh, look at that. In this lab it's 12 degrees now. It must be warming up. This is question 6 of the component 1 task book. We've got an experiment on preservation of food in the microbiology topic. We have got a graph, and we're storing shrimp paste. This is the start of the shrimp paste, and then as we store it over a number of days, so storing it for longer. And then we've got total viable count, so this is numbers of living microbes, and that's a log scale. We have control, which was no preservative at all. We've got then 2 other bars using 2 different preservatives, tea and ginger, and then together. Standard deviation, which is this spread around the mean, and then 2 other symbols just to show you what has made a difference. What is meant by total viable count? That's a nice straightforward start, which is the total number of bacterial cells. But it's 2 marks, so we're going to have to say 2 things. So first one, total number of cells or bacteria. And this is usually per something, so per mass, per volume, usually per volume, per volume of a medium or whatever you're testing. It could be per mass, possibly. Medium, you could have culture, substance, liquid. So just be careful with the 2 marks there. It's not just the total number of cells with 2 marks. Explain why we are using logarithms. Well, if we didn't use the logarithms, the graph paper that you'd need would be, from here into the next lab on the scale. You've got a large range and it'd be difficult to plot. So, first reason, large range of numbers. Particularly when these might be growing exponentially. Difficult to plot. So when you're using this scale, because each increase is a factor of 10, then you can squeeze that range down. Explain the importance of standard deviations and how the researchers used them to conclude there was no significant difference between the sample after 40 days. So 40 days, that's this one, and as we can see there, that's what we're talking about. Those bars there don't overlap, and that's the key bit of information there. So, importance of standard deviations. So explain the importance of standard deviations. That's the first job we're going to do. Standard deviation, just a definition, measures the spread of data around the mean. The standard deviation measures the spread of data around the mean. And if we were interpreting that for any question, that would be, the smaller standard deviation, the more reliable the data. The smaller the standard deviation, the more reliable the data. So we've explained there, either of those will do for that first mark. For this second mark, how would the researchers have used these to conclude that there's no significant difference? Well, we can see that these bars overlap. So there's overlap between the bars, and so there's no overlap between the standard deviations. There is an overlap between the standard deviations, the bars. And part D, using the information provided, what conclusions could you draw from the treatment of the shrimp paste with tea extract and ginger extract, and what advice would you give the best way to preserve shrimp paste? So this is a 4-mark question, so this is where a lot of students miss marks. So you need to break this down and think about where you're going to get these 4 marks. So using the information provided, we're going to have to use this graph. What could you draw from the treatment of shrimp paste with tea extract and ginger extract? Right, well, tea extract, that's our hashed line, and so we can see they, from say 80 days onwards, they've got a significant effect. So the tea extract alone, from 80 days, 120 days, and 160 days, yep, that has an effect. Ginger, that's our grey, then nothing at 40, 80 yes, 120 yes, 160 yes. So on their own we can say that they're more effective than the control. Let's write that down. So both tea extract and ginger extract, tea extract and ginger extract preserve shrimp paste. We could make the point that, well, up to 40, then there's no difference from the controls. So up to 40 days, no preservative, is effective. Up to 40 days, neither helps preserve shrimp paste. So we've talked about the 40, and then we've talked about the 80, 120 and 160 days. And we could even put some numbers on here, from 80 to 160 days. And then how about together? Well, together, what happens? Well, we actually get, from 120 days, we're getting the together extract working better than either of the single extracts. So this triangle here is saying it's significantly different to the single ones and the control. So from above 80 or from 120 days, at 120 days, the together extract, the tea plus ginger, is better than either alone and there is another mark. From 80 days, any of the 3 treatments are better than none. So that's another version of this point, really. Up to 40 days, neither helps. From 80 days, any treatment better than none. So sort of 5 things you could say there, and you just need to hit 4 of them. What further investigations would need to be carried out before you could use tea and ginger extracts to preserve shrimp paste? I don't know whether you like the taste of tea or ginger, but that might be one of the considerations. You might just want your shrimp to taste of shrimp. So the first, possibly the most obvious is the effect on the taste. So you'd need taste tests. Now, is the flavour affected? And then some people might be, affected by tea or ginger, might be allergic, something like that. So that might be a consideration. So allergies, toxins, that sort of thing from tea or ginger. And in these things, there's 2 marks, but you could always talk about cost effectiveness, and then all sorts of sensible things you could say. From 160 days onwards, you might want to test different temperatures, standard preservatives, and that sort of thing. So test other preservatives and so on. 12 marks, but quite difficult to get those 4 marks.